Potassium, or kalium, is a positive ion, or cation, noted with a K. About 98% of total body potassium is found in the intracellular fluid, or the ICF for short, which makes for an intracellular potassium concentration of about 150 milliequivalents per liter. The remaining 2% is in the extracellular fluid, or the ECF, which consists of plasma and interstitial fluid. However, since we can only measure the plasma level of potassium, which is about 4.5 milliequivalents per liter, that level is often used to define the normal extracellular concentration of potassium. Maintaining the normal potassium concentration in the ECF and ICF is essential for the normal functioning of excitable cells like nerve cells and muscle cells, including cardiomyocytes. Now, across all cell membranes, when there's no stimulus, there are negative electrical charges on the inside and positive electrical charges on the outside. This creates a potential difference called the resting membrane potential. Once there's a stimulus, like when a muscle contracts, an electrochemical impulse is generated and transmitted along the cell membrane, and that generates an action potential. Alright, now we get potassium from our diet. The daily recommended potassium intake is about 40 to 50 milliequivalents per liter, which is about 1.6 to 2 grams of potassium, which is the equivalent of 5 bananas per day. Once ingested, potassium is reabsorbed in the blood by the GI tract and travels unbound to plasma proteins. Most of potassium gets inside the cells. A little amount can be lost through the sweat and GI tract, and the rest is filtered by the kidneys and excreted. Knowing this, potassium needs to be carefully regulated in order for its concentration to remain constant. Potassium balance depends on the total amount of potassium in the body, which in turn is determined by potassium intake and excretion, and it's called the external potassium balance. Potassium balance also depends on the distribution of potassium between the ECF and ICF, and is also called the internal potassium balance. Alright, let's start with the external potassium balance. On a daily basis, the urinary excretion of potassium must be equal to the dietary potassium, minus small amounts of potassium that can be lost through the sweat or GI tract. Now, if potassium excretion is less than potassium intake, then a positive potassium balance and hyperkalemia, or increased potassium levels in the blood, can happen. If potassium excretion is greater than intake, then this is a negative potassium balance and hypokalemia, or low levels of potassium in the blood. So the kidneys have a pretty important job of keeping the potassium balance at a cozy neutral. Now, the kidneys are made up of lots and lots of nephrons, and each nephron is made up of a renal corpuscle and a renal tubule. The renal corpuscle, in turn, is made up of the glomerulus, which is a tiny clump of capillaries, and Bowman's capsule surrounding it. So blood gets to the glomerulus through the afferent arteriole, which is a branch of the renal artery, and leaves the glomerulus through the efferent arterioles. These vessels act like a coffee filter, allowing everything but red blood cells and proteins to pass from the bloodstream into the Bowman's capsule, which is connected to the renal tubule, and the resulting fluid is called filtrate. Now, upon exiting the glomerulus, the efferent arterioles divide into capillaries a second time, forming the peritubular vessels, which wrap around the segments of the renal tubule. The proximal convoluted tubule, the U-shaped loop of Henle, which has a descending and ascending limb, the distal convoluted tubule, and the collecting duct. As filtrate passes through the renal tubule, ions like potassium are filtered from the capillaries into the lumen of the tubule, and reabsorbed from the lumen into the capillaries, depending on the amount of potassium in the bloodstream. First, potassium is freely filtered across glomerular capillaries and moves on in the proximal convoluted tubule, or in the PCT, where 67% of the potassium is reabsorbed. Interestingly, 67% of the water is also reabsorbed in the PCT, leaving the lumen full of all kinds of solutes, like potassium. As a consequence, some potassium gets dragged passively from the lumen into the PCT cells and then into the bloodstream. Some of the potassium can also simply get dragged along with water, and this is called solvent drag. Further on, in the thick ascending limb of Henle, an additional 20% of the filtered potassium is reabsorbed using the sodium potassium chloride cotransporter, or NKCC2. Some medications, like loop diuretics, can block the NKCC2 cotransporter, and this results in increased sodium, chloride, and potassium excretion. Finally, the distal convoluted tubule and collecting duct are responsible for adjustments in potassium excretion, specifically adjustments when dietary potassium intake varies. So if a person has a low potassium diet, then more potassium will be reabsorbed in these segments. Conversely, when there's a normal or a high intake of potassium, more potassium will be secreted by the principal cells of the distal and convoluted tubule into the lumen. This is mostly regulated by aldosterone, a hormone produced in the adrenal glands in response to angiotensin II, which in turn is a part of the renin-angiotensin-aldosterone system that typically responds to low blood pressure. Aldosterone increases sodium and water reabsorption to bring blood pressure back up, and this also results in increased potassium secretion. So first, aldosterone induces the synthesis of more sodium channels on the apical surface of the distal tubular cells, as well as the collecting duct cells. This allows for more sodium to enter the cells, so more sodium that can be used for the sodium-potassium ATPase, found in the basolateral surface of the tubular cells. The sodium-potassium ATPase is a pump that gets 2 potassium ions inside the cells, and pumps 3 sodium ions outside the cells. As a result, sodium is pumped out of the cells and eventually into the bloodstream, while potassium enters into the cells. Aldosterone also increases the number of sodium-potassium ATPases, and as a result, even more potassium will be pumped into the cell. In this way, the intracellular potassium concentration increases, and this creates a driving force for potassium to be secreted from the cell into the lumen. Additionally, aldosterone also increases the number of potassium channels in the luminal membrane, so even more potassium gets excreted. Finally, another regulatory mechanism is the amount of negative ions in the distal tubule or in the collecting duct. When there are more negative ions in the lumen, like bicarbonate, compared to positive ones, potassium secretion increases to maintain the balance between positive and negative ions. Alright, let's switch gears and talk about internal potassium balance, or the distribution of potassium in the ECF versus the ICF. Internal potassium balance is mainly kept in check by the sodium-potassium ATPases on the surface of all cells. And most factors that regulate the internal potassium balance do that by acting on the sodium-potassium ATPase. So let's say an individual eats a huge amount of potassium, like 10 instead of 5 bananas. Well, all that potassium reaches the GI tract and gets reabsorbed in the blood, which could lead to a dangerous rise in the extracellular concentration of potassium. Fortunately, after eating a meal, a lot of insulin is secreted to help with the uptake of glucose into cells. And insulin also increases the activity of the sodium-potassium ATPase, so more potassium gets inside the cell as well. Next, catecholamines like epinephrine also increase the activity of the sodium-potassium ATPase, allowing more potassium to enter the cells. This is particularly important during exercise. On the other hand, during exercise, potassium tends to exit the cells. And that's because during exercise, we consume a lot of energy that's normally stored as ATP. Burning ATP for energy opens potassium channels, and in this way, potassium can escape the cells. Because of this, during exercise, there's a very small increase in extracellular potassium concentration, which helps the skeletal muscle arterioles dilate, allowing more blood flow to the muscles. But since epinephrine levels also rise a bit during exercise, that limits the amount of potassium that gets out of the cells, which maintains the balance. Another way for potassium to leave the cells is through cell lysis, which is when the cell membranes are damaged and broken down. This can happen with burns, rhabdomyolysis, which is when the skeletal muscle is damaged, or when cancer cells are destroyed using chemotherapy. When cell membranes break down, this basically leaves a door open for potassium to get outside the cell, leading to hyperkalemia. Finally, potassium balance is very responsive to changes in serum osmolarity and pH. Osmolarity reflects the number of solute particles per liter of solvent, and normally the osmolarity of the ICF equals the osmolarity of the ECF, even though the exact composition of solutes differs. Now, when there's hyperosmolarity, this means that there's something in the ECF that creates an osmotic force capable of dragging water from inside the cells, like too many sodium ions. As water leaves the cells, the intracellular potassium concentration increases, and this creates a driving force for potassium to leave the cell, leading to a rise in extracellular potassium. On the other hand, pH reflects the concentration of hydrogen ions, and normal blood pH is about 7.4. To maintain pH balance, hydrogen moves in and out of the cells. In order for hydrogen to move across the cell membrane, it must be accompanied by an anion, meaning an ion with a negative charge, or must be exchanged for another cation, like potassium. Now, when there's a primary decrease in the hydrogen concentration in the blood, this is called metabolic alkalosis. As a coping mechanism, hydrogen must leave the cells in exchange for potassium. More potassium gets inside the cells, and this leads to hypokalemia. Alternatively, when there's a primary increase in the hydrogen concentration in the blood, this is called metabolic acidosis. As a coping mechanism, hydrogen must enter the cells in exchange for potassium. More potassium leaves the cells, and this leads to hyperkalemia. Alright, as a quick recap. Potassium is a cation that's mostly located inside the cell, and it's essential for maintaining the function of excitable tissues. Potassium needs to be carefully regulated in order for its concentration to remain constant. The internal potassium balance is a distribution of potassium across cell membranes. Insulin and epinephrine increase potassium uptake by cells. Exercise, cell lysis, and hyperosmolality can increase potassium concentration in the ECF. Acidosis can increase potassium levels in the ECF, while alkalosis can decrease it. The external potassium balance is represented by the renal mechanisms that maintain potassium in balance, and as a general rule, the amount of potassium ingested must be equal to the potassium excreted. And finally, the distal convoluted tubule and collecting duct are responsible for adjustments in potassium excretion.
Sodium is a positive ion, or a cation, noted with Na. Most of the sodium in our body is located outside the cells in the extracellular fluid, or ECF for short. In the extracellular fluid, sodium has a concentration of about 135 milliequivalents, MEq, per liter. And remember that sodium concentration doesn't necessarily reflect the total amount of sodium in the body, but rather the amount of sodium relative to the amount of water in the body. So, sodium homeostasis refers to the mechanisms employed by the body to maintain a normal sodium concentration in the extracellular fluid. Sodium is essential in maintaining water balance, as well as for nerve impulse conduction and muscle contraction. Additionally, sodium is an important determinant of the volume and osmolality of the extracellular fluid, which is made up of plasma and interstitial fluid. Now, osmolality refers to the total solute concentration in a certain amount of solvent or water. By affecting plasma osmolality, sodium determines plasma and blood volume. So at the end of the day, it's important to maintain the sodium concentration in order to keep enough blood inside our arteries. This blood is called the effective arterial blood volume, or EABV, and it's what ends up perfusing our various organs and tissues. Okay, now sodium comes from our diet. The daily recommended sodium intake is about 2.3 grams per day, which is the equivalent of a teaspoon of salt per day. Once ingested, sodium is absorbed in the blood by the GI tract and travels through the bloodstream unbound to plasma proteins. At the other end, some sodium is eliminated from the body through sweat and through feces, but most of it comes out, along with water, as pee. So the kidneys are the cornerstone of sodium homeostasis. See, the kidneys are made up of lots and lots of nephrons, and each nephron is made up of a renal corpuscle and a renal tubule. The renal corpuscle, in turn, is made up of the glomerulus, which is a tiny clump of capillaries, and Bowman's capsule surrounding it. So, blood gets to the glomerulus through the afferent arteriole, which is a branch of the renal artery, and leaves the glomerulus through the efferent arterioles. These vessels act like a coffee filter, allowing everything but red blood cells and proteins to pass from the bloodstream into Bowman's capsule, which is connected to the renal tubule, and the resulting fluid is called filtrate. Now, upon exiting the glomerulus, the efferent arterioles divide into capillaries a second time, forming the peritubular vessels, which wrap around the segments of the renal tubule. The proximal convoluted tubule, the U-shaped loop of Henle, which has a descending and ascending limb, the distal convoluted tubule, and the collecting duct. As filtrate passes through the renal tubule, ions like sodium are filtered from the capillaries into the lumen of the tubule and reabsorbed from the lumen into the capillaries, depending on the amount of sodium in the bloodstream. So first, 67% of the sodium in the tubule lumen is reabsorbed in the proximal convoluted tubule, or in the PCT. This segment is also permeable to water, so whenever a sodium molecule is reabsorbed, water is reabsorbed along with it, which is called isosmotic reabsorption. Now, in the early PCT, sodium is reabsorbed together with other molecules through 3 different channels found on the surface of tubular cells. Sodium and glucose are reabsorbed together through the sodium-glucose cotransporter. Sodium and amino acids are also reabsorbed together through the sodium amino acid cotransporter. And finally, phosphate and sodium are reabsorbed together through the sodium phosphate cotransporter. An important regulatory mechanism here is parathyroid hormone, or PTH, which is produced by the parathyroid glands in response to low serum calcium or high serum phosphate. PTH inhibits the sodium phosphate cotransporter, so more sodium and phosphate are excreted. Finally, in the early PCT, there's also a sodium-hydrogen exchanger, which is a cell membrane protein that reabsorbs sodium in exchange for hydrogen. And this is mainly regulated by a molecule called angiotensin II, which is a product of the renin-angiotensin-aldosterone system. Now, renin is an enzyme that's released by the kidneys in response to hypotension. In short, the way it goes is that renin stimulates angiotensinogen conversion into angiotensin I, which is then converted into angiotensin II. Angiotensin II has many functions, some of which include vasoconstriction of the efferent renal arteriole and stimulating the sodium-hydrogen exchanger. In turn, this increases sodium reabsorption and water reabsorption in order to bring up blood pressure. Second, in the late PCT, sodium is still reabsorbed through the sodium-hydrogen exchanger and also along with chloride through the chloride-formate exchanger. This transporter reabsorbs chloride and secretes formate, which is a negative ion derived from formic acid. In the late PCT, sodium and chloride can also get reabsorbed through a paracellular way, meaning that they don't use any channels, but rather they sneak between 2 epithelial cells and go back into the bloodstream. If there's a lot of chloride in the tubules, then some of it will diffuse from the tubular lumen through the tubular cells and then in the bloodstream. Losing all those negatively charged chloride ions makes the lumen more positively charged than the bloodstream, creating a lumen-positive potential difference. This potential difference will drive the paracellular reabsorption of sodium as well. Okay, next, about 25% of the sodium is further reabsorbed in the thick ascending loop of Henle, or TAL for short. This segment, however, is impermeable to water, so no water is reabsorbed along with the sodium. In the TAL, sodium is reabsorbed, along with potassium and chloride, through the sodium-potassium chloride cotransporter, also called NKCC2. An important regulatory mechanism here is antidiuretic hormone, or ADH, which is secreted by the posterior part of the pituitary gland up in the brain in response to decreased blood volume or increased blood osmolality, so when there are too many solutes compared to water in the blood. When secreted, this stimulates NKCC2, increasing sodium, chloride, and potassium reabsorption. Next, in the early distal tubule, 5% of the filtered sodium is reabsorbed, and just like TAL, it's impermeable to water. Here there's a sodium chloride cotransporter that reabsorbs both sodium and chloride. In the late distal tubule and the collecting ducts, the remaining 3% of filtered sodium is reabsorbed. These segments are in charge of fine-tuning blood sodium concentration. The key players here are some cells in the tubule wall called the principal cells, which have epithelial sodium channels, or ENAC for short, on their surface. Sodium is reabsorbed by ENACs in response to aldosterone, which is the final product of the renin-angiotensin-aldosterone system. Aldosterone is secreted in response to low blood pressure, and it induces the synthesis of more ENAC channels, which allows for more sodium to be reabsorbed, so blood pressure increases. And just in case all that wasn't enough, there are a couple of other mechanisms acting on the kidneys to regulate sodium concentration and keep EABV and blood pressure in the normal range. First off, there's the sympathetic nervous system, which activates when there's a decrease in blood pressure and causes vasoconstriction of the afferent arterioles in the glomerulus. Additionally, it also increases sodium reabsorption in the proximal tubule. At the other end of the spectrum, there is atriopeptin, or ANP, which is secreted by the atria when EABV is increased, so high blood pressure. ANP causes vasodilation of the afferent arterioles and vasoconstriction of the efferent arterioles, which increases the glomerular filtration rate, so more blood is filtered by the glomerulus. ANP also inhibits ENAC channels in the late distal tubule and collecting ducts, decreasing sodium reabsorption. So more sodium and water are eliminated, decreasing blood pressure. Other peptides, similar to ANP, include urodilatin, which is secreted by the kidneys themselves, and the brain natriuretic peptide, or BNP, which is secreted by the brain and the cardiac ventricular cells. They are also secreted in response to high blood pressure and act just like ANP to bring it down, but they have a lower intensity. Let's finish off with two examples. When an individual has a diet high in sodium, the extracellular fluid and, in turn, the EABV and blood pressure increase. So atriopeptin is secreted, and the kidneys excrete more sodium and water, lowering blood pressure. Alternatively, on a low-sodium diet, the extracellular fluid, EABV, and blood pressure are low. In response, the sympathetic nervous system and the renin-angiotensin-aldosterone system activate, increasing sodium and water reabsorption to bring blood pressure back up. All right, as a quick recap, sodium is a cation that's mostly in the extracellular fluid and is essential for maintaining water balance, as well as nerve impulse conduction and muscle contraction. Sodium is filtered across the glomerular capillaries. In the PCT, 67% of the filtered sodium is reabsorbed through the sodium-glucose cotransporter, the sodium amino acid cotransporter, the sodium phosphate cotransporter, and the sodium-hydrogen exchanger. In the TAL, about 25% of the sodium is reabsorbed through the sodium-potassium chloride, or NKCC2 cotransporter. In the early distal tubule, 5% of the filtered sodium is reabsorbed through a sodium chloride cotransporter. The late distal tubule and the collecting ducts reabsorb about 3% of the filtered sodium through epithelial sodium channels, or ENACs. Factors that stimulate sodium reabsorption include the renin-angiotensin-aldosterone system, ADH, and the sympathetic nervous system. Factors that stimulate sodium excretion include PTH and peptides like ANP.